Okay, I can see the numbers are still ticking upwards, but seem to be slowing down. So I think we have a quorum at least. So hello everyone, and welcome to the fourth annual lunchtime lecture series of the Pediatric Gene Therapy and Medical Ethics Working Group, or PGTME. My name is Emma James. I am head of medical and patient affairs, as well as the medical office at Encoded Therapeutics, a company developing gene therapy for pediatric CNS disorders. I am also a member of the PGTME, and our panelists will be introducing themselves shortly. We're really grateful to have you all here for our fourth year of hosting this event. We're always really impressed and inspired by the diverse group of attendees and thoughtful engagement we get from the community. PGTME is a project of the Division of Medical Ethics housed in the NYU Grossman School of Medicine's Department of Population Health. You can learn more about PGTME on our webpage, and Cara will drop the link into the chat. And you can sign up for our quarterly newsletter to keep up with our work. We're really excited to hear from today's speakers on the ethics and science of controls in gene therapy studies. If you're unable to attend any of this week's talks, we will be posting captioned recordings once the week has concluded, and we'll share the link with all of the attendees. Today's event will feature brief introductions by our panelists, followed by a moderated discussion. We've also allowed 10 minutes for audience of Q&A, and we encourage you to submit questions through the Q&A feature throughout the panel. We have also received some of the questions already received during the registration submission, and we'll try and wrap those into our conversation today. Without further ado, I'm going to hand it off to our panelists to introduce themselves and their work. So let's start with you, please, Suyash.
Emma, thanks very much for the introduction, and it's a real pleasure to be here. My name is Suyash Prasad. I'm a pediatrician by training with a special interest in pediatric intensive care. So my clinical practice was looking after children affected by severe, devastating conditions. I moved into clinical research about 20 years ago, principally working in a number of different companies, rare disease companies. More recently, over the past 10 years or so, I've been working in the field of gene therapy. I've had senior roles at a number of gene therapy companies, chief medical officer, head of R&D, and really excited to be a part of this conversation. So thanks very much, Emma, and the rest of the team.
Thank you, Suyash. Alison?
Great. Thank you, Emma, and thank you, everyone, for being here and for inviting me to speak on this panel. My name is Alison Brent. I am a veterinarian by training. I'm an internist and an interventional radiologist, and I've been working in medical device development and clinical trials in animals with clinical unmet needs and significant disease my entire career until my daughter in 2015 was diagnosed with a rare monogenetic neurodevelopmental disorder called Angelman syndrome. And I got very involved in the research of Angelman syndrome and joined the Foundation for Angelman Syndrome Therapeutics, or FAST, and became their chief science officer, really being tasked with leading the drug development ecosystem from proof of concept through IND-enabling studies and into first-in-human phase 1/2 clinical trials. And in doing so, was lucky enough to be part of the founding team at a company called Genetics Biotherapeutics, where we founded a company out of the nonprofit. And we advanced an antisense oligonucleotide ourselves through IND-enabling studies and into a phase 1/2 clinical trial in partnership with Ultragenyx Pharmaceuticals, which is now currently acquired by Ultragenyx and being run in the phase 1/2 by them. And I currently still advance the other disease-modifying gene-targeted therapies for Angelman, including many gene therapies, lentivirus, AAV gene therapies, as well as enzyme replacement therapy and CRISPR for the disorder. And I also serve as the chief development officer for a company working on other monogenetic neurodevelopmental disorders called Mazi Therapeutics. And I'm their chief development officer, really in charge of taking proof of concept and human candidate through IND-enabling studies for the first in human clinical trial. Thanks for having me.
Thank you, Alison. And Peter?
Hi, Peter Marks, Director of Center for Biologics Evaluation and Research at US FDA. We're the center that regulates cell and gene therapies in the United States. And I am a hematologist oncologist by training with background both in academic medicine and industry. Thanks.
Thank you. Thank you all for joining us. So fundamental to clinical research is the tenet that ethics must be a central component of research, design, and practice. And in order to be ethical, clinical trials must also be scientifically valid. However, attempting to balance the values of advancing scientific knowledge with protecting human patients can pose challenges, and potential conflicts between the standards of scientific research and ethics are notable with respect to trial design, and particularly with respect to the choice of control arm. Ethical dilemmas can be particularly conspicuous in early phase pediatric gene therapy trials that involve highly novel approaches and surgical procedures. This is a complex topic, and we will likely only scratch the surface in today's session. I want to acknowledge that we're extremely fortunate to have all of our panelists, and particularly for Peter joining us as a senior member of the FDA. At PGTME, we're grateful every time members of the FDA and other regulatory agencies agree to participate in these discussions, which require perspectives from science, bioethics, advocacy, industry, regulatory, and policy. And with that regard, I'd like to remind everyone that our hope is to make progress on these complicated issues. To do that, contributors need to be able to safely vet ideas that are informed by personal and professional experiences. Over time, some will stick, others will evolve, and hopefully we will advance the space together. So with this in mind, today we will focus our discussion on the clinical trial designs and appropriate controls for trials of AAV-based gene therapies for rare pediatric genetic disorders, and comments should be considered through this specific context. Before we jump into the questions, we thought it'd be useful to briefly explain the different kinds of controls and terms that we're going to be using, particularly as they apply to gene therapy trials, which Suyash will spend a few minutes on. So over to you, please, Suyash. And Aidan, if you could just bring up the screen.
Great, thanks, Emma. Thanks, Aidan. So I think Emma's comment about the fact that it's really important for the scientific design of the clinical trial to be able to demonstrate whether a drug works or not. That in itself is ethical. You know, it's important to design the study well in order to be ethically appropriate. Otherwise, you're putting subjects at risk. And I think there has been this central idea in clinical research for many decades now that ethical considerations have to be central to research, design, and practice. That's why we have IRBs. It's why we have the FDA. Despite the fact that this idea is clearly important, there's sometimes there's a balance between advancing scientific knowledge on the one hand and protecting human subjects on the other, especially when it comes to children, especially when it comes to highly innovative therapies such as gene therapy. And this conflict is especially salient when it comes to clinical trial design and the selection of a control. And I'm just going to go to high level through some definitions. Clinical trial methodology is very complex, but I'm just going to go through some very high level points that help add some context to the discussion. So you'll hear us talk about terms such as observational study, an observational trial. And if you look at the first row on this particular slide, you can see that we've got retrospective observational trials and prospective observational trials. And this essentially is a way of understanding the natural history of a clinical condition. And for rare and severe pediatric gene therapy diseases, often there isn't that much information around. And so it's important to understand the disease in order to be able to understand it, develop a therapy, and also design a clinical trial. You can look at observational data both retrospectively, i.e. you can look back by looking at patient case notes and looking at data. And with recent advances in data management, this is becoming easier and faster with high quality results. You can also look at it prospectively, where you design a study, you enroll children prospectively into this trial, and you observe them over time, and you look at various parameters, whether it's seizures or neuromuscular function or respiratory function. You look at these, the progression of these children over time to get a sense of how these children with a particular condition progress. And in fact, patients in this type of prospective study could actually enroll into an interventional trial at some future time point. Looking at interventional trials, you can see there's three buckets here. And there are many more, but these are the three that are most salient to this discussion. The first is an open label interventional trial. Open label means open, i.e. both the clinicians and the patients know whether they're on treatment or not on treatment. A parallel study is where you can have two arms, i.e. one group that's randomized onto treatment versus another group that's randomized onto placebo, or randomized onto non-treatment. And this can be both blinded or not blinded. And we'll get onto more of a discussion on that as this discussion progresses. You can also have a crossover study where patients are randomized either onto treatment or no treatment, that observe for a period of time, then they cross over onto treatment. If they've been on no treatment, they go on to treatment. If they've been on treatment, they then have no treatment. This is not quite relevant for a gene therapy study, because with gene therapy, once you've been dosed, you are treated. But there is a delayed treatment type of study which is related to this crossover design, which is relevant. Now, two important concepts that often get confused are this notion of blinding and the notion of randomization. They're actually two separate concepts, and they reduce different types of bias in the study, which you are trying to remove as much as possible to run a scientifically intact study. So randomization removes selection bias. It's where a group of patients are randomized, i.e. they're chosen randomly either to go into treatment or no treatment. This adds some statistical rigor to a study. Blinding is where, double blinding in particular, is where neither the patient nor the clinician treating the patient know whether the patient is on treatment or on no treatment. And this can become quite complex operationally, but is felt to be the best way of reducing what's known as reporting bias, i.e. if you know you're on the treatment, you may feel, the clinician may feel, maybe biased to actually scoring you higher in your clinical improvement than if you don't know whether you're on a treatment or no treatment. And the double-blind placebo-controlled study is the gold standard, theoretically, from a drug approval process. But as we'll find out, it's inherently challenging in the field of pediatric gene therapy studies. Next slide, please. And then you can have, the other way to look at this discussion is look at internal controls versus external controls. The internal controls are, you can have a placebo or a sham. A placebo is where you give something that looks like the treatment. It looks, feels, and appears like the treatment, to keep the patient and the clinician blinded, i.e. not knowing whether they're being given active drug or not. And there's a whole discussion around the integrity of the blind that we'll get into as well during this conversation. You can also have a no treatment concurrent control, i.e. people know they're not on treatment, the patients, clinicians know they're not on treatment, but they're still being compared to a group that are on treatment. And you can have this delayed treatment control where you are not treated and observed for a period of time, and then you get treated after 6 months or 9 months or a year perhaps. So those are the different types of controls that are typical in these gene therapy studies. Or you can have an external control, which is outside of the remit of the particular study, which is usually drawn from literature, scientific literature, or a retrospective observational study, as we talked about, or a prospective study also. And there are ways of making the external control as scientifically rigorous as possible that we can get into a discussion on. Okay, so those are some high-level thoughts on the types of study design that are pertinent to this discussion. So let me hand over back to Emma, and you can carry on with some questions.
Appreciate it, Suyash. Thank you very much. So to start off with the Q&A then, we have some sort of prepared questions that we thought would be a good introduction to the topic. And then, as we mentioned at the beginning, please do add other questions into the chat. So Peter, starting with you, at a high level, how do regulatory assessors define what is ethical for pediatric trials? And how does this shape their assessment of whether something is safe and effective?
So in the United States, we actually are driven by the Code of Federal Regulations in this regard, in that our Title 21 of the Code of Federal Regulations, Part 50, Subpart D, tells us that for additional safeguards for children in clinical investigations, research involving children either has to be restricted to minimal or minor increase over a minimal risk absent a potential for direct benefit to the child, or the alternative is that it has to present risks that are justified by the anticipated direct benefits to the child, the balance of which is at least as favorable as any available alternatives. And that's generally what we're thinking about when we're thinking about gene therapies for children. So for rare diseases, it often boils down to evaluating the scientific basis for the gene therapy, the preclinical data that are available, any data from adults, if there are data from adults, and seeing whether we feel like that risk-benefit is in the positive direction. Now, in some cases, there are no adult data because there are no adults to treat, in which case it's a matter of carefully looking over the preclinical data, the nature of the construct of the gene therapy, what it's targeting, what's being measured, and deciding whether or not everything is overall favorable. And sometimes it can be quite challenging because sometimes the animal models in these situations are not perfect. So we try to get it right as much as we can, but it is a challenging, can be challenging to decide whether there is prospect of direct clinical benefit sometimes.
Okay, thank you. And I guess, to follow on from that, Alison and then Suyash, it would be great to hear, does this assessment differ from the patient community and then also the sponsor community? And also, are you seeing any discrepancies between how regulators are viewing this versus, say, IRBs as they are assessing clinical trial protocols? So Alison, perhaps I'll go to you first.
Yeah, sure. I think that the language in the guidance and in the regulations is exactly right, and I think that is what families want, that is what caregivers want, that's what patients want, and certainly that's what communities and all stakeholders probably want. The question, I think, more is the perspective of how is it carried out and what is the interpretation of that language. And I think that's where there's some contradiction at times or some concern at times in the risk families, patients, and caregivers are willing to take in understanding risk-benefit calculus. And the risk of doing nothing can oftentimes outweigh in the lives of families living with these significant, debilitating, if not life-threatening diseases. The risk of doing nothing can outweigh the risk of doing something. If the package, the safety package, and all of the data is supportive of doing something and moving into a clinical trial for prospective benefit. And so I think that's where the challenge in the community is more so of where do we lie in this risk calculus? Because us doing nothing feels more risky than trying to do something. Understanding the community has to be very well educated, and those that are entering trials need to have a very, very clear understanding from the primary investigator who's consenting these families what the risks exactly are. And if that is there and they do understand that, then I think them being willing to take that risk, maybe regardless of their age, regardless whether or not there's anything known of whether they will miss a therapeutic window. That's unknown in these first in man trials of what is the therapeutic window. And mice can tell you so much, but the human story is very different. And I do think some folks making these judgments or these rules or making these statements don't necessarily live with the disease. And when you don't live with the disease, it is very hard to hear risk assessment by somebody who doesn't know what it's like to live with risk every day. And I think that's where patients need to be put at the center of the conversation, and patient-focused drug development, which certainly is a very coined term that a lot of people use, really has to involve that decision being a combination of regulators, families, caregivers, and patients, and industry, because I do think there's a very different perspective on what that risk is, depending on what angle you're looking at this from. And if we're all working toward the patients and the families, I think it's important that they're part of every conversation. And those conversations are between industry and regulatory more commonly than the patients and the families. And they're getting like the repercussions of the conversation, not actually being part of the conversation.
Yeah, yeah. And Suyash, from your perspective, is there anything else you would add from the sponsor perspective?
I mean, I would echo Alison's comment that in my experience of running gene therapy studies or studies in severe, devastating illnesses, parents and families are more willing to take risks than you might imagine. And getting this balance between the potential for benefit versus doing nothing is an important concept. I also think that there's a judgment piece here. You know, one of the conversations we often have in our meetings in the company is, okay, well, we're convinced ourselves that this drug works, but what do we need to do now to convince a regulator, the FDA or one of the European regulators? And sometimes the bar is different, and sometimes there's quite a bit of width. There's quite a bit of difference between those two bars, and you know, I don't think there needs to be. I think, you know, if we as clinicians and parents and families and knowledgeable experts feel, okay, this is enough information for a drug to be approved and go out there and benefit patients and families, while still also collecting data and more information on how the drug works within the context of the disease, there shouldn't be that much difference to what we feel and what the FDA or other regulators feel. So I think reducing that gap is particularly important, especially when you've got the operational complexities of running rare disease studies where there aren't that many patients around, there aren't many doctors who know about the disease, and especially with a very innovative new therapy.
Yeah, yeah. So Peter, I guess we've seen the FDA exercise regulatory flexibility, you know, in rare disease where mortality is a feasible endpoint or where the natural history is homogeneous in the outcomes, but not all diseases offer this clarity. So can you tell us a bit about how the agency weighs aspects of the disease, such as prevalence or natural history or the endpoints, to advise developers on the trial design and specifically the requirements for the controls for, say, first-in-human studies and as well as for evaluation for licensure?
Yeah, no, thanks. And obviously, I think what was just said makes is very true. Is that we want to work with the patient community to find what is acceptable, knowing that at the end of the day, though, that what gets through the process has to work. And I have to tell you, the one cautionary tale before I go through these different things is that sometimes there is such a desire to have something that we can, you know, we have to be sure that we at least set the bar at a certain place. And that's why we do have our scientific standards, and we take into account prevalence, natural history, endpoints. Just to give you an idea, for prevalence, for, you know, there, although we do sometimes require randomization in order to get the most rapid answers to questions, that often really is reasonable for larger populations. And we know for rare diseases, particularly for very rare diseases, randomization is either difficult or just frankly impossible. And so we instead have to, for these very low prevalence diseases, have to think about using individuals as their own comparator in terms of change from baseline, or historical comparisons, or sometimes run-in periods to look at clinical function while we then look at changes. We can look at natural history because well-defined natural history may help us avoid the need for randomized clinical trials, even in some larger populations of rare diseases and smaller populations of rare diseases. If you think about spinal muscular atrophy type 1, you know, because it was a more common rare disease, spinal muscular atrophy type 1, and the natural history was very well defined. No comparator was needed, so no randomized trial. But that said, there are very rare diseases for which we have little information about their natural history, or where very few individuals have the disease, or those that do have the disease have enough of a variable manifestation that it can be challenging. And that means that sometimes we have to, if we want to get there without randomization, we have to then fall back on using biomarkers that can be measured as changing during a treatment period, ultimately leading us towards accelerated approval. And that's something, though, that gene therapies are very well tailored to, because if a gene therapy is giving a particular is replacing a particular protein product, that can end up serving as an endpoint. So now we get to the endpoints, where really endpoints can come in different forms and are disease dependent. And in some cases, when you have a disease course that's rapid and consistent, clinical endpoints can prove useful. But more often, such endpoints are variable, or they take too long to occur. And so we end up needing to think about using biomarkers. And gene therapy, again, by replacing a protein that's deficient, or by knocking down a protein that's present in excess, you can measure what you have achieved and hopefully correlate that to either an animal model in which you can show that the level that you've achieved is associated with clinical improvement or absence of clinical manifestations, or you can look to the human experiment, because many times for some of these diseases, we have people in the population who have some abnormal level of the protein, too low, perhaps, that are without overt manifestations of disease. And if you can achieve that, again, another way of using a biomarker. Of course, we need to ultimately confirm those with clinical findings, but I think we're very much of the mind, and I think this is trying to be patient focused, that we'll use our accelerated approval provisions to lean in and try to get things across the finish line, initially using biomarkers as an endpoint, in order then to come back, if necessary, could even be a few years later, with the clinical data that confirms clinical benefit and transitions these to full approvals. For us, it's very important that we have enough evidence initially through a biomarker that we can get an insurance company to reimburse for these products, because this is a major issue, right? That if these products can't get paid for, they're not going to get to patients, and we've seen that time and time again.
Yeah, for sure. Could go lots of different directions here. I'm looking at the questions and answers here. I think I
can make a comment on endpoints if you like, Emma.
Oh, sure, go ahead.
Because I think this is an important point. When you have an endpoint where there's a big change, a big clear difference, the need for a placebo or a blind is much reduced. Okay, if you have a population that's very heterogeneous, or if you look at the converse rather, if the disease progression is very consistent, i.e. there's a lack of heterogeneity, then the need for a blind or a placebo is also reduced. If you're looking at an endpoint that is, to build on Peter's point, more mechanistic in nature rather than functional or subjective or effort dependent, then the need for a placebo is reduced. And I think these are all concepts, I'm sure, Peter, that you talk about in detail when you're reviewing IND submissions and clinical trial design. So for those of us that are designing studies, if we can rely on isolating a more heterogeneous population, if we can try and select endpoints that are going to choose a big swing and choose doses of drugs that are going to produce a big swing rather than a smaller change, that's important. If we can, to Peter's point, have a clinical endpoint that is probably the primary endpoint, but heavily supported by key secondary biomarkers or mechanistic endpoints, I think all those things contribute to the ability to have a study which does not have a placebo, does not need to be blinded.
And just to add to that as well, I totally agree. And I think we have to define and really understand the difference when we're talking about a phase 1 clinical trial and a pivotal phase 3 clinical trial. And even though these are rare diseases, we're held to those standards, of course, for scientific rigor to have to do that. And I think a lot of where the ethical question lies are the first in human. And in order to understand if we have a true therapeutic benefit, and if that benefit is very strong, that treatment effect is strong, you have to actually understand how the gene therapy behaves in the population. And being that most of these diseases have never seen a single treatment and have no comparator, you don't know how things are going to behave. Are, nonverbal individual is going to start speaking, or non-walking individual is going to start walking? That's profound. Or is it going to be that activities of daily living are going to change, or there's going to be a very clear biomarker change that are going to be explored in that phase 1, phase 2 trial? So I think it's really important that we have to think about the disease. We have to think about the heterogeneity of that disease. We have to think about, is survival the benefit we're going for, or is it quality of life change, independent living change, or we actually aren't sure, but the way they're living currently is unacceptable. And therefore it's a global disorder that we want to see profoundly change in many ways, but we don't know what's going to change first. We don't understand the development of the brain or certain body systems at certain ages. And I do think the exploratory nature of a phase 1/2 shouldn't be underestimated. And that's where when feedback is given for a first in human to do a sham control or a placebo control, I think that's more of a pushback than it is a pivotal understanding. Now we have some idea of where we're going in a pivotal. Can we justify a randomization in a control arm versus not, because it's too small of a population or the change is so great and we don't need it? So I think those two are very different conversations.
Yeah. So Alison, I guess, how does the patient community, assess the difference of the risk between that first in human versus the confirmatory studies? Do we need to do more to make sure that they understand sort of the difference in the kind of the evidence requirements required at the different phases?
That's a really great question. And, I can obviously speak for the Angelman community, and we take that very seriously as a patient advocacy group. That is our job, to educate our parent stakeholders, right? There's a lot of stakeholders in our community, and education of families to understand what's coming and really repetitively have them participate in experience surveys and perspective surveys, and really understand the disease concept and what is a minimal meaningful change and what is a placebo, what is a control, what is a sham. Education's everything. And I would say that our community repetitively is educated on this, and they're ready. And so they understand that they may need to be in a control arm, and they understand, and in a non-degenerative disorder are willing to accept that in a situation in which there's no harm. So for a gene therapy, if they have to go on immunosuppressive therapy and be in a control arm, they would consider that harm. Versus in a small molecule control arm where it's a true placebo and there's no harm in that placebo, then they would have less of a concern, as long as it's short-lived and as long as there's prospect for benefit that they will cross over. And it's a short enough time that they're not missing a potential therapeutic window. But again, we don't know what that is. So I think it's all about education. And I find that the responsibility of the patient advocacy groups to do a really good job of that. And then second to that is the PIs in these trials to really educate their patient community in terms of enrollment. And then at the advocacy group meetings where pharmaceutical companies come and present their program from an early stage, they need to talk about this, because repetition for non-medical people is incredibly prudent. And they're very smart. They're more sophisticated about this disease than you will ever be, than anybody at the FDA or in industry will ever be. They understand risk. We can't underestimate the knowledge and the sophistication of these families. They're itching for knowledge. They want to understand. And so we give it to them. So they're ready when we come in to say, this is why you need a placebo. But if you don't explain why, and you just go and you just say, this is how it's going to be, that's unacceptable for communities. And as long as they're really educated on the why, again, if it's a degenerative disorder, and by the time the placebo is over, a part of the population might have passed, that would be a totally different conversation. And that's a different ethics, conversation to be had.
Yeah, it's a great point. And one of the questions here, again, I guess to Alison, to you again, So what do you think patient-focused drug development means when you have a patient community that might be willing to try anything? This is one of the questions we have in the chat. Like, how should the patient community's willingness to try play into the regulatory decisions? And I guess maybe, Peter, if you have any comment to make on that after Alison, that'd be great.
So how do parents, just say that one more time?
So I've just lost the question. It basically, how should the patient community's willingness to try anything feed into regulatory decision making when you incorporate the concept of patient-focused drug development?
Yeah, so I don't think parents are willing to try anything. I think that's an overstatement. I think that's that's not true. I think parents are willing to try things that they're educated about, that they can see a prospect of benefit for. They can understand the mechanism of action that it can help their child. They can, or their, an adult or a pediatric child. That they can see that the alternative, what the alternative is compared to that. And parents need to know what all of their options are. And I think that a lot of parents are not willing to just do anything, but they're certainly willing to take a big risk for a big reward. They're willing to take a small risk for a small reward. But they're not willing to take any risk for any reward. And there are some families who are very risk intolerant. And that's okay. And we educate that everything is okay. And we really want parents to feel comfortable asking the right questions. And some do not want to be in a phase 1 because they don't want to be an open label. They don't want to be the test child to get the high dose that could be toxic or the low dose that could be ineffective. And now they have no other opportunity down the line. So that's a risk of being in a phase 1-2 trial that's open label. And then the risk of placebo is the phase 3, if you have a placebo arm or a control arm, is not getting anything. And that's a big risk. So all of these things are risks. And there are families that are very willing to be in a phase 1 trial. There are families that are waiting for phase 3 because they want to know, at least if their kid gets a drug or a gene therapy, that it's the best possible dose. And then there are families that are just willing, I would say it's, a third. And then there are families that are wanting to wait for an approval and they just don't want to take the risk in a trial at all. Or they want to be in something like an ASO because it's reversible versus a gene therapy, which is not, because we have both options. So I really think it's all education, and families are not willing to just do anything. I think as long as they're educated, they're going to make really smart decisions.
Yeah, I would just follow up with that and say I agree completely that my major concern, and I think our major concern, is that accurate information is provided to those parents. I think the times that I've seen kind of the worst instances, most egregious episodes that I've seen since being at FDA have been when either inadvertently or intentionally claims have been made that are overreaching about what a product can do. I think provided parents are given information about what the possible benefits are, potential risks, and uncertainties around a product, they will determine what suits them. And there really is a range. I think Alison said it very nicely. There are a range of risk tolerance, just like people have risk tolerance in investment, ranging from people who need an absolute certainty on their investments to other people who are willing to take tremendous risk. The same thing goes actually with medical care. And I think it's okay, provided people have accurate information on which to base those decisions. And so from my perspective, I think from the regulator's perspective, it's very important that we're very clear about the information that goes out there. We will never let anything through that we think is wildly risky. Okay, I mean that's our job, is to make sure that stuff that is potentially dangerous doesn't make it out there. But on the other hand, there's always, especially as we move into the realm of more complex gene therapies, there's going to be some risk involved, and our job is to make sure that that risk is well taken, calculated, and that the parents and to the extent that they're older patients are informed of what those are.
Thank you, Peter. And we have a ton of questions on, I guess, the effectiveness of different types of controls. So, so the clinical trial of gene therapies obviously often require steroids as well as invasive sham procedures, and it can be questionable as to whether effective blinding is really possible. What's your view of that functional unblinding within a clinical trial? Are there acceptable ways to generate robust data without a full blind, for example? And then people are asking, for example, can we talk about the ethics of a sham procedure in light of CFR 50.52, which is clinical investigations involving greater than minimal risk but presenting prospective direct benefit? And what kind of endpoints, if you had a double-blind, double study in a rare disease gene therapy, could you just in fact have randomization if the endpoints were clear? Would that be sufficient for registration? So maybe if you could just talk a bit about how you think about some of that, and then we can ask Alison and Suyash too.
Yeah, so let me just back it up to say that we encourage randomized trials when randomized trials make sense. And that is when populations are sufficient size, when they can be interpreted appropriately, executed appropriately, and when they actually get to the answer in the most forthwith manner. And that is it can be challenging to get to an answer in some diseases for which we don't have perfect understanding of the disease, or for which we don't have good biomarkers. That said, for many of the diseases that we're talking about that are very rare, randomization is, we acknowledge randomization is not going to be possible. It's just not possible. And the point is well taken that sham controls in pediatric populations, the ethics of those, we could spend the rest of the day going over here about whether they really are reasonable to do, and under what size procedure is reasonable to do as a sham. But the idea here, hopefully, for most of the time for gene therapy, and I'm going to stick with gene therapies for the moment, is that if you really understand what your intervention is, right, that you are either filling something that's empty or knocking down something that's in excess, you can hopefully find a way towards using a biomarker to figure out what you have done and whether what you have done matches up with something that, at least in a model system or in an unaffected human situation, matches with essentially an improvement in a person's condition, or with lack of absence of disease manifestation. So the idea here is that you're trying to use the biomarker to avoid this need for randomization, to get across the finish line under our accelerated approval provisions. Because for diseases where there are 100, 200 individuals in the United States or less, the idea of randomizing it's very challenging, and it also it's also potentially delays the ability to get these studies done, because oftentimes parents are not very excited about having their children randomized. It's sometimes, I think, going back to the basic science, spending the time getting the animal models to work, or doing the pre-work to show correlations with humans who have reduced levels of a given protein. Doing that up front can sometimes save a lot of time out back, because then you can run a single-arm trial, use accelerated approval based on a biomarker, and get across that finish line much sooner than were you to have to try to randomize, which is very challenging. It's the point is not lost that in some cases it's almost like pseudo randomization, because although you've done the randomization, because of effects associated with products. Sometimes those who have received the product know what they've received, and very quickly people figure out on the trial who's gotten what. So better to try to get that sorted out and not to have to use randomization if you can get there in a manner that can get compelling data that the product actually likely has activity.
So you mentioned biomarkers several times over the course of today's panel. And one of the questions is that this works well for things that are measured in the blood, but how do we think about this when the proteins are only expressed in, for example, the brain?
Right. So this gets to some challenges of having to deal with whether you have to look at CSF biomarkers, which is doable through lumbar puncture, spinal tap. It's the most challenging when you don't have any fluid that actually expresses the biomarker. And then you're left with this situation of either looking at a functional endpoint or potentially having to do biopsies. And in the brain, that's not something that's very exciting to be doing. That's where we tend to have conversations with developers to look at what makes the most sense. And it's very hard to make a generalization here that fits every development program, because in some cases, even though one biomarker may not change, the one you're actually intervening on, something else may change that you can measure, and either measure through something chemically, or sometimes there are radiographic changes that can occur, and we're open to any type of way of showing a difference has occurred. That could include use of MRI scans, PET scanning, etc. So that helps us get some window into issues with brain function.
Peter, can you comment on your how you interpret EEG for neurodevelopmental disorders? There's a huge push toward, individuals with monogenetic neurodevelopmental disorders have some significant EEG findings that animal models can show change with therapy over time. How do you view that as a biomarker?
Yeah, it may turn out that that's a very reasonable biomarker, especially because using AI, EEGs are a bear to read. And you can imagine a lot of EEG data could be very challenging, especially if you have it, continuous EEG monitoring over 24 hours or longer. But I think that's a place where AI may help us, right? Because you could potentially have baseline EEGs over the course of days, and then post-treatment EEGs at various time points. And then even though humans would probably tear their hair out if they were ever assigned to do that even for one individual, AI could probably do that for multiple individuals and get to some of it. So I think we're very open to these, particularly because I think now with some of supercomputing, we can actually put it to good use. I think everyone's very worried about the bad things that supercomputing can do, but we've been using supercomputing for years in our center for different things. And here's, I think, one of the issues where supercomputing can potentially benefit. And I would encourage developers thinking about something like this to come in and talk to us early on, because I think this is a case where sometimes pilot studies to make sure that we're getting the right, being able to discern the right changes could be helpful.
Yeah, I would add another comment to the use of biomarkers, but we talked about two different types of biomarkers here. One is a simple blood-based number that we get, where there's not much interpretation. But then, to Alison's point, EEGs do have some interpretation, and biopsies, for example, have some interpretation. And one of the ways that you can handle that or add some scientific rigor is in a biopsy or in an EEG, you could actually blind the assessors. So you don't have to blind the study, you don't have to blind the investigators or the patients, but you can blind the assessors of the EEG. You can say, you can give 3 electrophysiologists, here are a bunch of EEGs. We're not telling you who's on drug, we're not telling you who's not on drug. You can even throw in a bunch of dummy EEGs as well from miscellaneous patients. And that's a way of adding some scientific rigor to the interpretation of some of these biomarkers, which I think is really important. And I think some of this flexibility is increasingly being shown. I mean, I think there are a number of examples where I think folk have been upset with the FDA being too rigorous, but more recently, I know, and a good example was Grace Sciences LLC, who had an AAV gene therapy IND approved for NCLY1, a rare metabolic disease, and they've agreed to a single-arm open-label study with both a clinical functional endpoint, the Bailey, and also a biomarker, GNA. And I think these clinical functional endpoints can also be, you can add more rigor because you can videotape things like the Bailey or the Denver Developmental Scale or the CHOP Intend, and you have them assessed by the clinician assessing them, but then also you can have them double scored by blinded raters in a different part of the country. And that, once again, it's not ideal because sometimes you need to be there in person doing the assessment, but it's another way of adding more integrity in a blinded manner without blinding the patient or the treatment or the clinician to treatment, and without needing to use a placebo.
Thank you. I can see we're almost running up out of time again. We have a bunch of questions also, I guess not specifically to sham, but Suyash, maybe I'll ask you. So sham is one aspect of blinding, but can we also talk about the burden, risk, ethical considerations between adequately blinding a gene therapy study with prophylactic and immunosuppression, and along with the frequent blood work that should be done in control subjects? Like, how can we minimize that risk to people who are having controls?
Yeah, I mean, this is where it becomes, this is where you start to lose the integrity of the blind. This is where functional unblinding happens, because if you give a gene therapy to a patient, we all know there's a risk of LFT elevations in the first few weeks or some kind of immunological consequence, and you have to take lots of blood tests frequently to try and head that off at the pass before the LFT elevation rises too high. So especially in children, there's limits on how much blood you can take. So if one group of patients are receiving the immunosuppressive therapies and having lots of blood tests, that's fine. But then if another group are not receiving the immunosuppressive therapies, you're still doing lots of blood tests. It's not really, it doesn't feel comfortable. That's arguably not an ethical thing to do. I mean, the way to blind immunosuppressives is you make a placebo immunosuppressive as well, which adds complexity and cost and delays timelines. But you can't not, you're going to be unblinded if you're doing less frequent blood tests in that group as opposed to the gene therapy treated group. So it becomes a really challenging question, especially when there are prescribed limits on the number of blood tests, the amount of blood you can take from a pediatric patient. So, I would not be in favor of a blinded study, especially in very young children, where you have to take lots of blood tests. It's not that ethical. You could also argue that biopsies, for example, or repeat lumbar punctures are also, it's inherently uncomfortable and potentially unethical to do that in an untreated patient where they are not receiving benefit. And sometimes it's a judgment call. Often it's a judgment call.
I would also say that steroids, even if you're using a placebo, like a placebo steroid, steroid rage is something that children certainly get. And you will know a child on steroids or a child on a placebo of steroids in a minute if you've ever had a child on steroids. So I think you will inadvertently unblind if you don't have them all on steroids, because you will know immediately if your child within a week is on steroids or not.
One of the questions, we have a lot of questions here. Sorry, everyone, we're not going to get to them all, but I'm trying to filter through them. So gene therapy trials by necessity surface ideas for more efficient controls. How do you see innovations in controls and study designs in gene therapy impacting the broader clinical trial landscape? Could we expect adoption across other therapy areas, small molecules, biologics, more common diseases?
You know, I'm going to say this. I'm not sure that I'm not sure that we're going to be able to leverage everything we can do with gene. I'm very happy to be able to use the uniqueness of gene therapies and the idea that that if you actually know the structural protein or enzyme that is missing, that you can put it back in and measure it. And unfortunately for small molecules, we're not always able to do that. Right now, I'd love to just see us leverage the heck out of this, and get more things across the finish line with accelerated approval. And then maybe we can start thinking about how it could possibly apply to small molecules, et cetera, where it's going to be more challenging because you don't have that direct, I mean, think about like a reductionist, to me, the reductionist case as a hematologist is, you know, hemophilia. You have a missing clotting factor. We know that when you get to a certain level of that clotting factor, you don't bleed. So basically, if you can give back that clotting factor to a functional activity of a certain amount in an individual, you know they are not going to bleed. You have a biomarker. And unfortunately, you can't do that same thing with a small molecule. Can't do it quite the same way with an antibody just at this point.
No, I would say that there's definitely, I agree with you, Peter. I don't think you can really translate some of these approaches to small molecules. But the way gene therapy is evolving, it's not just gene replacement therapy anymore. You've got AAV packaged CRISPR, AAV packaged RNA, AAV packaged targeted protein modifications. And I think what we learn from gene therapy can morph and can influence some of those technologies where you're marrying gene therapy with another approach. So I think there's definitely room for thinking and translation there, but not to small molecules or some of the older ways of doing things.
I just want to build on that to say that I think what we will see is what we have already been doing with AAV is going to translate very nicely to CRISPR as we, and probably CRISPR will go even further as we have to, we'll have to use ways of really having platforms to be able to have multiple different RNA guides, be able to move forward. So I think we're really kind of, I think you're right on target with that.
So I have two last questions. Alison, I'll ask you to go first, and then I have a closing one for Peter. But Alison, what would you like to see from regulators when discussing study designs in rare disease drug development? More clarity, consistency, flexibility, something else? How can we move this forward?
Yeah, I mean, I think there's already been really nice changes that we've all seen. So I would say staying on that same trajectory of just balance and flexibility. And so for me, you know, I've heard Emil Kakkis use the word equipause. So like we want to think about the regulatory equipause and the patient equipause and industry. And the fact for me is that I find it, now having lived the industry world for some time, I'm finding that the amount of interaction we have from the industry side with the regulators in helping us to make these decisions, and the amount of interaction we have from the patient advocacy side with trying to understand how these decisions were made, and everyone kind of coming around the table together to have a bigger conversation for every program, which is a huge ask. But I think it actually helps solve a lot of the issues of getting around the why. And I think, you know, industry has this mindset that they don't want to get put on a clinical hold. So they'll kind of do whatever the FDA says because they just don't want to get on a hold and they don't want to take a risk and they just want to get through. And so they'll bend more than maybe they should because they feel ethically it's not right. Versus the patient advocacy groups are not seeing that side of it. And they're just like, why? And if the regulatory agencies had a bigger conversation with industry and patient advocacy, they may have said, you know, you're right. You don't need to include a control in this. And this is why. And I agree with you. But I feel like the engagement is so minimal for many reasons that it makes it really hard. And I would say more engagement and putting the patient at the center of the conversation in the center between industry and regulatory, not out on the outskirts hearing about it later, because I do think it would make a better relationship for everyone in terms of ethics.
Appreciate it. And Peter, I guess building on that as our last question, How can sponsors, IRBs, patient community better influence the agency and specifically the reviewers to express their views on the potential challenges that this scientific rigor may pose? You've talked very publicly about the changes in the leadership of the FDA and what you consider to be important changes to advance gene therapy development, but can you share how you envision this cultural shift to roll out over the next year or two, and how it may then we can impact how individual applications are assessed kind of across the board?
Yeah, I think just to build on what Allison said, I think we are headed in a direction very much where Allison is hoping that we'd head, which is that I think we are increasingly going to have focusing on the patient as kind of the center of what we are trying to do. That is what Nicole Verdun, who's the new head of the Office of Therapeutic Products, the area that handles gene and cell therapy in our center, is doing. I think what we probably will try to work toward is more of this incorporation of listening to patient advocates, potentially having meetings where workshops where we have both patient advocates and industry involvement, and also just having our reviewers try to have more of a sense of what patients are living with or their families are living with, because sometimes that has been very compelling for them, not just hearing what it's like being a patient with a disease, but also hearing how much goes on to support those individuals with a given disease. And in fact, that is, I think, in some cases, the one of the better kept secrets of rare diseases, is that for many rare diseases, although the individual with the rare disease suffers tremendously, the burden on the families is tremendous. And when our reviewers hear that, they sometimes, it helps them put things in perspective. So I think you'll see us trying to focus on the patient and their family in the context of the disease ever more so.
Thank you. And with that, we are out of time. I wish we'd had a full day, I think, to get through all of the questions that we have. We will try and put together some information and answer the rest of the questions that have been posed in the Q&A, and get back to people on that. I would like to thank our panelists for a great discussion today, and for all of the attendees for all of those thoughtful questions. Be sure to tune in for tomorrow's panel, which is the antibody question and how does it impact trial enrollment. And on behalf of PGTME, I would like to thank everyone who's attended, participated, or helped put this webinar together. Thank you, and have a great rest of your day.
Thank you, Emma.
Thank you.
Thanks.
Bye-bye.